In chemical synthesis, click chemistry is generating substances quickly and reliably by joining small units together. Click chemistry is not a single specific reaction, but describes a way of generating products that follows examples in nature, which also generate substances by joining small modular units. The term was coined by K. Barry Sharpless in 1998, and was first fully described by Sharpless, Hartmuth Kolb, and M. G. Finn of the Scripps Research Institute in 2001. A desirable click chemistry reaction would be modular, be wide in scope, give very high chemical yields, generate only inoffensive byproducts, be stereospecific, be physiologically stable, exhibit a large thermodynamic driving force to favor a reaction with a single reaction product. A distinct exothermic reaction makes a reactant spring-loaded, have high atom economy. The process would preferably have simple reaction conditions, use readily available starting materials and reagents, use no solvent or use a solvent that is benign or easily removed, provide simple product isolation by non-chromatographic methods. Explanation: proteins are made from repeating amino acid units, and sugars are made from repeating monosaccharide units. The connections are carbono-euro-heteroatom bonds CXC, rather than carbono-euro-carbon bonds. In addition, enzymes ensure that chemical processes can overcome large enthalpy hurdles by a series of reactions each requiring only a small energy step. Mimicking nature in organic synthesis may facilitate the discovery of new pharmaceuticals given the large number of possible structures. In 1996, Geiger calculated the size of the pool of drug candidates at 1063, based on the presumption that a candidate consists of fewer than 30 non-hydrogen atoms, weighs less than 500 daltons, is made up of atoms of hydrogen, carbon, nitrogen, oxygen, phosphorus, sulfur, chlorine and bromine, is stable at room temperature, and does not react with oxygen and water. Click chemistry in combination with combinatorial chemistry, high-throughput screening and building chemical libraries speeds up new drug discoveries by making each reaction in a multi-step synthesis fast, efficient and predictable. Many of the click chemistry criteria are subjective, and even if measurable and objective criteria could be agreed upon, it is unlikely that any reaction will be perfect for every situation and application. However, several reactions have been identified that fit the concept better than others. 3+2 cycle additions, such as the hydrogen 1,3-dipolar cycle addition, in particular the Copper-catalyzed stepwise variant, are often referred to simply as click reactions, THIOLEne reaction, D-else order reaction and inverse electron demand D-else order reaction, 4+1, cycle additions between isonitriles and tetrazenes, nucleophilic substitution especially to small-strained rings like epoxy and aziridine compounds, carbonyl chemistry like formation of ureas but not reactions of the aldol type due to low thermodynamic driving force. Addition reactions to carbon-carbon double bonds like dihydroxylation or the alkanes in the THIOLEne reaction. Azide-alkane-hydrogen cycle addition. One of the most popular reactions within the click chemistry concept is the azide-alkane-hydrogen cycle addition using a copper catalyst at room temperature. It was discovered concurrently and independently by the groups of Valery V. Fokin and K. Barry Sharpless at the Scripps Research Institute in California and Morten Meldal in the Carlsberg Laboratory, Denmark. Although the Copper-catalyzed variant was first reported by Meldal and co-workers for the synthesis of peptidotrizoles on solid support, these authors did not recognize the potential of the reaction and did not make a connection with the click chemistry concept. Sharpless and Fokin independently described it as a reliable catalytic process offering an unprecedented level of selectivity, reliability, and scope for those organic synthesis endeavors which depend on the creation of covalent links between diverse building blocks. Applications: click chemistry has widespread applications. Some of them are two-dimensional gel electrophoresis separation, preparative organic synthesis of 1,4-substituted triazoles, modification of peptide function with triazoles, modification of natural products and pharmaceuticals, natural product discovery, drug discovery, macrocyclizations using Copper-catalyzed catalyzed triazole couplings, modification of DNA and nucleotides by triazole ligation, supramolecular chemistry, calixarenes, rotaxanes, and catenanes, dendrimer design, carbohydrate clusters and carbohydrate conjugation by Cu-1, catalyzed triazole ligation reactions, polymers and biopolymers, surfaces, material science, nanotechnology, and bioconjugation, for example, azidocoumarin. Biomaterials, click chemistry has also been used for selectively labeling biomolecules within biological systems. A click reaction that is to be performed in a living system must meet an even more rigorous set of criteria than in an in vitro reaction. It must be orthogonal, meaning the reagents used may not interact with the biological system in any way, nor may they be toxic. The reaction must also occur at neutral pH and at or around body temperature. Most click reactions have a high energy content. The reactions are irreversible and involve carbon heteroatom bonding processes. An example is the storing and ligation of azides. Technology license: the Scripps Research Institute has a portfolio of click chemistry patents. Licensees include Invitrogen, Allozyme, Aileron, Integrated Diagnostics, and the biotech company BasicClick, a BASF spin-off created to sell products made using click chemistry. Moreover, BasicClick holds a worldwide exclusive license for the research and diagnostic market for the nucleic acid field. Fluorescent azides and alkanes also produced by such companies as Active Motif Chromium and Shiand. References. External links: Click Chemistry, a review, Click Chemistry, short review and recent literature, National Science Foundation, feature Going Live with Click Chemistry. Chemical and Engineering News: feature In situ click chemistry. Chemical and Engineering News: feature Copper-free click chemistry, metal-free click chemistry review, Click Chemistry, a ChemSoc Rev themed issue highlighting the latest applications of click chemistry, guest edited by M. G. Finn and Valerie Fokin. Published by the Royal Society of Chemistry.